Good morning. Welcome to Monday, everybody. Just wanted to go through this slide deck with you a little bit as you begin your Principles of Heredity study. There is, of course, first of all, a good tie-in to the cells unit because there's a guy by the name of Gregor Mendel in this unit, and we'll talk about him in a little bit. This is just the home screen, and of course, the next screen shows you what you can write in your lab book if you should decide to do anything there. Moving right along, we come upon a guy by the name of Gregor Mendel. And there's lots more information. If you would like to click on this more about Mendel, you can find out some more things about him. But Gregor Mendel was the father of genetics. And he was a monk, which means he slept in a room that was called a cell. Exactly. Some of you already were there before I got there. But he was the gardener at the monastery that he lived, and he noticed something about the pea plants that he planted at the monastery. He noticed that some pea plants turned out to be tall, and other pea plants turned out to be short. So he was a curious monk, and being the gardener and he decided to do a little study on pea plants. So he did a small science project that lasted for 7 to 10 years because planting peas, you can only really plant one generation of peas per year. So he did a study where he, first of all, continued to plant pea plants that were tall, peas from pea plants that were tall. And he kept planting them in the garden until finally every pea plant in that garden was tall. He did the same thing with short pea plants. He planted short pea plants and took the peas and planted more short pea plants and more short pea plants until all the pea plants were short. Then he took the pea plants and he cross-pollinated them, which means he took the pollen from the tall ones and he pollinated the short ones. And he took the pollen from the short ones and he pollinated the tall ones. He planted them in another garden. And what he found out was all the pea plants in that garden were tall, believe it or not. He then took those pea plants and he self-pollinated them. He took the pollen from the same pea plant and he pollinated with itself and found out when he planted those pea plants that not all the pea plants were tall. So tall plus tall equaled tall. Short plus short was always short. But when he took tall and short, he got tall. And when he took those talls and crossed them with other talls, he got mostly tall, but not all. Now, we're going to learn a little bit more about that when we talk about Punnett squares near the end, but that was his main experiment, and that's why he's called the father of genetics. Made a little mistake here. I'll be right back. There we go. That's why he's called the father of genetics. Phew. All right. Interesting. Let me try this again one more time. That's why he's called the father of genetics. There we go. Genetics is the study of heredity. You've already seen that, hopefully, in your vocabulary. Heredity is when you pass traits from parents to their offspring, from one generation to the next. That's why you have all the traits that you have. Some of the main principles he came up with was that traits are passed from the parents to their kids, to their offspring. And that there are little things, he called them factors, but now we call them genes that determine the specific traits that an organism is going to have. And you get one from mom, and you get one from dad. Now, there are strong ones and there are weak ones. And in the pea example, the tall was the strong, and now we call those dominant traits. And they hide the weak ones. So if there is a tall gene, it doesn't matter if there's a weak gene. The plant is going to be tall. It would take 2 short genes for a short plant to appear. So that's what it says here. A dominant gene hides a recessive gene. So there are these things called alleles, and if you listen to my song called the Heredity song, you'll hear about alleles, and maybe I'll sing it a little bit for you. But genes are represented by alleles, and alleles are basically letters that represent the genes. So if we're talking about pea plants, we can talk about the dominant capital T, and we can talk about the recessive lowercase t. Those are the alleles. If you put alleles together, you make up something called a genotype. According to the song, 2 alleles together make a genotype. 2 alleles together make a genotype. Whoa, here I go, floating up to get out of the text. If you look at the genotypes, those genotypes then determine what the creature is going to look like, what the characteristic is going to be, whether it be tall or short pea plants, whether it be long or short antennas, whether it be tall or short in terms of other creatures, whether you have anger or not. Those are all some of the characteristics, some of the phenotypes that result from the combination of genes that you have. So there we see the definition of allele: dominant being strong, recessive being weak. Genotype, when you put two letters together. You can see there's two different types of genotypes. There's homozygous and heterozygous. Homozygous, meaning they're the same, and heterozygous, meaning they're different. Another name for homozygous being purebred, and another name for heterozygous being hybrid. Now, there's two different types of homozygous. There's homozygous dominant and homozygous recessive, because if you just say homozygous, you're really not saying anything, because homozygous means same. And you can have same where they're both tall, and you can have same where they're both short. So you have to be very specific when you talk about homozygous. There we see homozygous and heterozygous and hybrid. Now, the phenotype is the way something looks or acts sometimes, depending on what you're talking about exactly, but the trait that happens. So that brings us to this whole idea of something called Punnett squares. And I'm going to float right over here. Isn't it nice to be a floating bubble? I've always wanted to be a floating bubble. Okay, that's a perfect place. Punnett squares are produced by somebody by the name of R.C. Punnett. And he created them to determine how traits get passed from one generation to the next. So you can see here is a Punnett square. Here are the male gametes, the male alleles, the genotype. Here are the female gametes, the female alleles, the genotype for the female. And here are the possible outcomes for the kids. Now, I'm going to go ahead and make one for you so you can kind of see how this works. So the first thing you always start with on a Punnett square, if I can get myself out of the way, there we go. First thing you always start with on a Punnett square is you start off with the key. So in our example, the key is going to be capital T for tall and lowercase t for short. Then you start with, or you continue with, what are the traits that we're crossing? What are the genotypes of the parents? So our first parent is going to be capital T, just like the pea plants that Gregor Mendel planted in the tall garden. The second parent is going to be lowercase t, just like the little plants he planted in his garden. So we're going to take these, and I make this colorful just for this one, just to show you so you can see where these letters go. But you can see we're going to take this black T right here, and we're going to put it on the top part of the box because that's the first parent. Now, according to the example on the page before, it said that the top is the male and the side is the female. It doesn't matter as long as one parent is on the top and the other parent is on the left side. The second T, here's how we did it this time. So that black T then fills down into these two boxes. These are the possibilities of what the kids' traits might be. The second T, the red T, goes across that box and fills in below just like the black T did. Moving on to, so that's the first parent. It goes across the top and its letters fill in down. The second parent, the little t, the blue T fills in here and fills in across. The green T fills in here and fills in across. Now you're going to see there are 4 different possibilities for what the offspring is going to be in this cross. And in this case, all 4 of them are the same. Big T, little t, big T, little t, big T, little t, big T, little t. But in a Punnett square, what we do next is we look into each one of those boxes and we determine whether it's going to have the dominant trait or whether it's going to have the recessive trait. And if it has the dominant trait, like that one does, we shade it somehow. Pencil, you can use the bucket fill, whatever that you can make it. But don't cross out the letters because you need to see them. This one also has the capital T in it. It's going to be tall, as is this one, as is this one. So all 4 of them turn out to be tall. Another way you can look at it is you fill in this box in the upper left-hand corner by looking up and to the left. Here, up and to the left, up and to the left, up and to the left. So since none of them are unshaded, that makes our ratio be 4 tall to 0 short. So that's how our ratio works. That's a mathematical thing that you can learn from Miss Kelly or Miss Jet sometime, or you can just learn it from me. That's what a ratio is. The last thing we do is we look then over here and we fill in what % chance does from this cross is it that the kids will be tall or short. And of course, it's going to be 100% tall and 0% short. in this particular case. Now, when you make another one, you use whatever letters are here. So if this is big T, little t, then this would be big T, little t. If this was big T, little t, you'd put a big T here and a little t here. So you change the letters depending on what the letters are that are there. So if you have questions about that, please make sure you ask, because you're going to be making a whole bunch of those in this unit of genetics. Now, these are just a couple of the different possibilities of how it can turn out, and you can look over these on your own at some point. These are some questions that are important, and if you're doing the present mode and you click, the answers are going to appear right here underneath me. And so just watch carefully that you know the answers to these questions, because these are some of the basics of heredity. And finally, the last thing is make sure you write down a few questions and some things that you still would like me to cover, because that way when you come, if you come to the help desk, you will have an idea of some of the information you would like to ask me. Questions? See me later, and hopefully you understand a little bit more about genetics.